students. Good morning. Welcome to the sessions on general microbiology. This is Kaushika Patel, Assistant Professor from LG Institute of Pharmacy, Ahmedabad. And today we are going to study about the different methods for the quantitative estimation of the bacterial growth. In the previous sessions, we understood what is a bacterial cell and what is the difference between bacterial cell and eukaryotic cell. Now, when there is a growth in the bacterial cell, how we can measure the growth rate in case of a sample of bacterial cells. So let's begin with our session. Now, this quantitative measurement of bacterial cell, they are also known as bacterial counting methods. Okay, so the same topic may be referred to as bacterial counting method. So there are different methods by which you can calculate the number of bacterial cells present in a particular sample. Before that, what is the growth of bacteria? Bacteria divide by binary fission method. In this method, in binary fission, a parent cell divides into two ideal daughter cells. Both these daughter cells have the same set of genes present in the parent cell because DNA replication occurs before the division of cell. Now, the multiplication rate of bacterial cell is very high. There is exponential growth of bacterial cells with respect to time. So normally when we are saying the growth, we refer to increase in size. But in case of bacterial cell, it is increase in the magnitude of the cell. That means the increase in the number of bacterial cells will be referred to as the growth of bacteria. Okay, so the different methods are there for counting the bacterial cells. These methods, they are of two types, either total count methods or viable count methods. Now, what is the difference between these two important terms? When we say viable count, it means a count of the living bacterial cells which can multiply exponentially and they will develop colony when inoculated on the solidified culture medium. Each bacterium can give rise to a single colony on the surface of the culture medium. So, viable count is measured in terms of CFU per ml, that is colony forming units of bacteria per ml of sample. Now, when we are going for total count, we are counting the total number of bacterial cells in the sample, irrespective of whether those cells are living or dead. So that is represented as total number of cells per ml of the sample. So what are the different methods for bacterial counting? Three different approaches are used. First is determination of cell count or directly cell number. Second is the determination of cell mass or weight. And the third is the determination of cell activity. For each of these concepts, you can have direct method or you can have indirect method for that approach. So each of this method you have to study in detail, and in exam you have to elaborate only those methods which are asked or only those approaches which are asked to you in the question. So first approach, that is determination of cell count. If we are counting the cell number by direct methods, or that is known as total count method, we are counting both the living and dead cells present in the sample. For that purpose, we have two different methods, direct microscopic method and electronic counting method. Okay, so we will see both the methods one by one. First, direct microscopic count. In this method, you have to use a microscope. To prepare the sample, you need a counting chamber like Petrov-Hauser chamber, or you can even use a Newbers chamber which is used to calculate the white blood cells in the blood sample. Okay, you have to prepare the slide by pouring the diluted sample on the slide, and then you have to observe the number of cells present in the special ruling of those slides, either by staining the sample or by using phase contrast microscope. Why? Because bacterial cells, they are transparent. You cannot view them directly. So you have to either impart color to the cell or you have to impart color to the background to create a contrast, and then you can view the cells present in the sample. You have to do the calculation and then you can count the number of cells present per ml of the sample. Advantages is it is very simple and easy method. You can get total count of bacteria directly. Along with the number of cells, you can also study the morphological characteristics of the cell like shape, size, and arrangement. Any type of dense sample can be processed by suitable dilution of that sample. The dilution factor you have to consider in the calculation at the end. But now, what are the limitations? You cannot distinguish between viable and non-viable count. If the sample is too much diluted, that too less number of cells are present, then it will become difficult to find the cells. Or if the cells are very small or they are motile, then it will become difficult to observe them in the microscope. Second method for counting the total number of cells is electronic counting method. This method is very accurate and easy compared to the microscopic method. And you can also get the size distribution range of the cells in this method apart from the number of cells. You can process this method for any type of particles apart from bacterial, virus, or any type of cell you can process in this device. Now, what is the principle? As you can see in the diagram, there will be a cell counter. Okay, in this counter, electrolytic solution is placed. There will be two compartments, anode and cathode, which will be separated from each other by a small orifice. This aperture is of 10 to 30 micrometer in size. Now the sample is added in this cell counter, and when the bacterial cells passes through this orifice, there will be increase in the resistance between the two different compartments. This increase in resistance will generate an electric signal which will be recorded on the screen. The number of bacterial cells is directly proportional to the number of electric signals which are recorded on the screen. So you can directly get the number of cells present in the sample. Now, what are the advantages? It is same as microscopic method, except that you cannot study the morphology in this method. But what are the limitations? Again, you cannot distinguish between viable and non-viable count. The most important thing is the sample must be homogeneous. Each and every cell should be dispersed uniformly. Otherwise, the clumps or aggregates will together recorded as a single electric signal, and there will be error in the result that you will get. You have to maintain the device properly because the orifice may get clogged. You have to clear it from time to time. It will require a sophisticated equipment, so the processing of the sample will be costly compared to that of microscopic method. Now, the second type of methods that are the indirect methods for the cell count, they are also known as viable count methods. In this method, you will count only the living cells which are developing into colony in the form of CFU per ml. Okay, again there are two methods, plate count method and membrane filter method. In plate count method, you have to inoculate the sample on a plate by a suitable method. You have to develop the colonies after a suitable and incubation period. The number of colonies obtained on the plate should be between 30 to 300. Such plate will be taken, calculated, and CFU per ml of sample is obtained. Now, this method is widely used because it is very easy, sensitive, and it is versatile because different type of samples like food, water, or milk can be processed by using this method. Now, plate count method is further of two types. You can go for pour plate method or you can go for spread plate method. The general advantages of this method, as I said, is it is very easy and sensitive method. It can be used for any magnitude of population because you can do dilution up to 10 raised to 8 times. So by suitable dilution, you can process any type of bacterial number using this method. But now, what are the limitations? Again, only viable count can be obtained. Dead bacterial cells will not be counted. You can count only those bacteria which are capable to grow on inoculation on the culture media to develop the colony. If the cell is present but it is not developing colony, then you cannot use this method. And again, the sample should be homogeneous. Otherwise, the entire clump will form a single colony and you will get error in the results. So now the first method, pour plate method, what you have to do in pour plate method, you have to use liquefied melted nutrient agar. You have to inoculate the culture sample, the culture of bacterial sample into that at 45 degrees Celsius. You have to roll the tube. You have to pour it in the sterilized empty Petri plate. Allow that plate to solidify at room temperature. Incubate for 24 hours at 37 degrees Celsius. And then you have to count the number of colonies after 24 hours, either manually or by using electronic counter. Now, the disadvantage is that you cannot operate the sample which is thermosensitive because you have to carry out the process at 45 degrees Celsius. The second spread plate method is better because here you don't have to use any higher temperature. So it is suitable for all the types of bacteria, even thermosensitive bacteria. Now, what is the difference? In spread plate method, you have to use directly solidified nutrient agar plate, which is sterilized. You have to inoculate the sample on the plate by spreading it using a spreader. And then you have to incubate it at for 24 hours at 37 degrees Celsius. And after that period, you have to count the number of colonies. Okay. Now, the second method for viable count is membrane filter method or membrane filter count. Now, this method is a modification of plate count method itself. But what is the difference? You will use this method when the bacterial load is very less. Plate count method is used when the population is dense and the sample size is small. Here, you have a membrane filter method is used when the bacterial load is very small and the sample size is large, like air. You want to measure the number of bacterial cells present in a room, in an empty room. Then the cells number will be less compared to the sample size. So then you have to go for membrane filter method. In this method, you have to pass the sample through a membrane filter of uniform pore size having predetermined pore size, and then the entire membrane along with the cells which are filtered, you have to inoculate on the culture medium, allow the colonies to develop, and then count the CFU per ml. Second approach is the determination of cell mass or cell weight. Here also there will be direct methods and indirect method. In direct method, you are directly determining the dry weight of the cells. Here you will filter the cells, you will allow them to dry, and then you will find out the weight of those cells. Now, what is the problem in this method? You should take care that only cells are present in the sample and any extraneous matter generated by the bacterial cells is not present. Otherwise, the weight of those intracellular material will also be counted in the total weight of the cells. Second is the determination of nitrogen content of cells. Now, this is an extension of first method or the modification of first method. Here, after you have separated the cells, you have dried them, you will determine specifically the nitrogen content of the dry cells. Why? Because it is believed that 14% weight of the dry cells is that of nitrogen. So, you will analytically measure the nitrogen content and then determine the number of cells present in the sample. Now, what are the limitations here? You should have a concentrated population. That means the dense sample should be there. Diluted sample cannot be operated. It will require a sensitive method and a sophisticated instrument, so it will be costly. And third, no other source of nitrogen should be present in the sample, or that will also be counted in the total nitrogen content of the sample. Second important method is indirect method for cell mass counting, that is the turbidimetric method. This method is very important from exam point of view also. Now, here you have to measure the turbidity of the sample. Okay, so inoculation is done in liquid culture medium. In plate count method, we require solid culture medium. In turbidimetry method, we require liquid culture medium. You have to measure the turbidity of the sample. Higher the turbidity of the sample, more will be the number of bacterial cells present in it. Okay, so you will require either spectrophotometer or colorimeter or turbidometer. What is the principle? In the sample compartment, you have to fill your suitably diluted sample. The light is allowed to pass through monochromator, and that single beam of light will fall on that sample. That beam of light will be scattered by the number of cells present in the sample, and that rays will be detected by the photocell detector present on the other side of the sample compartment. You have to develop a linear relationship between the turbidity and the number of cells per ml. Okay, and that way you have to generate a standard curve. Now, by using that standard curve, you can determine the turbidity of any unknown sample. What are the advantages? Again, it is very simple and accurate method, and you can process it for the bacterial suspension with up to 10 raised to 7 to 10 raised to 8 cells per ml. But what are the limitations? When the number of cells are less, this method cannot be preferred. So it is preferred for dense suspension but not for dilute suspension. If you are using colored liquid medium for growing the bacterial cells, then that color of the medium may interfere with the turbidity measurement of the sample. And sometimes what happens, any other sample, any other material present in the sample will be considered in the turbidity measurement, and so you will not get the accurate count of the number of cells present in the sample. Third approach, that is the determination of cell activity. Here only indirect method will be there because you will count the number of cells based on the biochemical activity of the sample. Okay, so you have to consider any end product or metabolic product or any chemical which is produced by the cell during the biochemical cycle. Okay, more the quantity of that material, higher will be the number of bacterial cells present in the sample. So like this, indirectly based on the biochemical activity of the cell, you are determining the number of cells present in the sample. Okay, so these are the three different approaches to count the bacterial cells in a sample. Different types of questions are asked in the exam. Okay, so methods for viable count, methods for total count, they are very important. You can even ask the differentiation between viable and total count. Then four methods of the beginning, that is the two methods of viable count and two methods of total count, they are very important. And apart from that, you may be asked turbidimetric method for the determination of cell mass. Okay, so all these five methods you have to study in detail, and you have to write properly in the exam with respect to what is asked in it. Okay, so I hope you have understood the different methods for the determination of cell mass. In the next lecture, we will start with the other topic related to the growth of the bacterial cell, that is growth curve and the qualitative estimation of the bacterial growth. Till then, take care. Subscribe on our YouTube channel Pharma Ignite for the more informative sessions on pharmacy. Thank you, goodbye, and see you soon.